think we can start. So, good morning, everyone. Thanks for coming. Thanks also for the people online. I hope you hear us. Let us know if you have any questions regarding technical issues on the chat, and if you hear us well. Today we have Thibault Stalder giving us a presentation. I would like to ask Nyanya to introduce him.
So it's a great pleasure for me to introduce Thibault. Thibault is a microbiologist with a strong interest in microbial evolution and ecology. Uh, he did his PhD at the University of Limoges, where he works on how antibiotic resistance spread in the environment. He then moves to US to join the Evatops group, when he mainly worked on the role of plasmid in the dissemination of antimicrobial resistance. And a couple years ago, he came back to France, where he was awarded an junior internship, and he's now passed again at the University of Limoges. And he currently worked on the mechanism driving the emergence of mechanism of the emergence and the spread of antimicrobial resistance, particularly through horizontal gene transfer across environmental, animal, and hospital ecosystems. So, Thibault, floor is yours.
Well, thank you very much for this introduction, and thank you first very much to Diva and Stephanie for the invitation, and it's very exciting to be here, meet them, and we have a lot in common, so it's a really good conversation, and in general, it's very exciting to meet the community in Montpellier that's work on ecology and evolution. So as Diva explained, so I'm a researcher at the Resinfit team at the University of Limoges. It's an INSERM team where we are mostly interested in antimicrobial resistance in general, with people working on viruses and some other bacteria. And so my current research is basically trying to understand what are the hub of antimicrobial resistance transmission across the One Health continuum, and the presentation I'll do today is a little bit more about the path, the trajectory that brought me to these questions, and share with you some of the latest results also. No, wrong one. Yeah. So first, maybe for people that are not microbiologists, why antimicrobial resistance? So AMR is a major global health issue with more than 1 million people dying directly from the consequence of AMR globally per year. And what is interesting about AMR is that the bacteria and their resistant genes do not know ecological border. And so this issue actually goes beyond what we would imagine from the human and the animal that are the most affected by antimicrobial resistance. But actually, what now we start to know better is that the environment play a major role in the emergence, dissemination, and transmission of AMR. So we really need to have this so-called One Health approach if we want to try to understand a little bit what's happening in terms of antimicrobial resistance. And so what we know, and has been shown over and over now over the last 15 years, is that first, AMR is ubiquitous, meaning that we find it everywhere. If we take samples from everywhere in the environment, we're always going to find antimicrobial resistance. And it's been shown pretty well over the last 15, 20 years. And what has been also well described, and here I just put a long list of the publication from the INSERM Resinfit team that contribute to that, but there's many people all around France and the world that show that is that anthropogenic activity tend to enrich for AMR, meaning that, for example, if we look in a hospital effluent, we'll find a lot of AMR, it's going to be very abundant. And if we look in the more environment, we'll find it, but it's much less abundant. And as we go through here, for example, a river with different impacts from, let's say, wastewater treatment plant, what we found is that often wastewater treatment plants will have a significant impact on the enrichment of antimicrobial resistance downstream those wastewater treatment plants. And what's been also well demonstrated is that on farmland, soil that are fertilized with organic matter, such as like slurry or basically slurry and manure, are also tend to be enriched with antimicrobial resistance. And another thing that has been well known now is that environment is actually a reservoir for those antimicrobial resistance. And so that's kind of like a little bit like what's been described. So for example, that's link us to is there is like a risk of the environment for the health of human and the animal when we talk about antimicrobial resistance. We'll say yes, it can be a problem because there's two well-known examples that have been well described where the environment play a role of like origin of certain resistance that we found in today pathogens that are giving a lot of trouble in hospitals. So the first one is, for example, the resistance to an antibiotics, the beta-lactams, that is encoded by a gene, CTXM. And it's been well shown that this gene originated into bacteria from aquatic environment previous, and then those genes would have been mobilized on what we call mobile genetic elements that ended up on some plasmids. So plasmids are those circular DNA elements that can that allow those resistant genes to move horizontally between different bacterial species. And these genes ended up, this plasmid, this gene ended up in certain E. coli that spread out throughout the world and give a lot of issue in terms of like antimicrobial resistance at the hospital. The other well-known example that we witnessed around 2015 is the colistin resistance. That was kind of a colistin is an antibiotic that was not much used in for human because it has a lot of side effects. But it was one of the part of the first antibiotic discoveries, maybe I think in the 50s or 60s. And so people started to use it again because there's like more and more multi-drug resistant bacteria, and sometimes there is like therapeutic, sorry, hard word to say, therapeutic dead hand where the doctor have to use some drugs that haven't that may be more efficient. So those are called last drug resorts, and colistin is one of them. But as soon as people started to use this drug, boom, some resistance pop up like in the strains, and they're mostly carried by plasmids. And so what has been shown is actually colistin has been used a lot as a growth promoter in Asia for poultry and swine industry. And so very likely from based on study, the same thing happened is that it's been identified that in Southeast Asia, those genes, so another type of gene, so the MCR1 gene, got mobilized from the background chromosome of bacteria into mobile genetic elements. Those mobile genetic elements then spread out throughout the world and now like are disseminated across many different bacteria. So what I'm interested in understanding is a little bit what are the eco-evolutionary trajectories that basically bring those genes on their mobile genetic elements, specifically plasmids, to move between different ecosystems. And so here, I just give you an example where the environment impacts the human or the animals, but there's other pathways here that have been studied a lot, where we have high environment where we have high selective pressure for certain type of resistances, and then through waste, those will eventually reach the environment, whether if it's by wastewater or manure, and often it's mixed up with residues of complex cocktail of pharmaceutical, such as antibiotics. And so we don't really know what happens when this bacteria also gets released into the environment, if those bacteria associated with the human and animal will die, or if they're going to have the time to give their resistant gene and plasmid to bacteria from the environment, creating new reservoirs. So there's a little bit both where we have kind of like things happening in the trajectories. And so what I try to do is trying to get this multi-level scale approach where I try to understand the fate of antibiotic-resistant genes. And so at either the level of the molecule, so here looking very specifically understanding in this context plasmids, but also how those plasmids adapt and evolve within their host population, and then how those resistant genes and plasmids also evolve and their ecology in bacterial community, and in more general have this overview of the roots of transmission of antimicrobial resistance on this one Earth continuum. And so today, I will focus mostly on the work that I did on the ecology, what I call the ecology of resistance plasmids in the environment. I'm not going to show the work that I did more on the evolution here, as I thought that you already saw that piece of work when at the SFM when we met, and so I was like, well, I'll show something different. So here it is. And so one of the main questions I had a little bit over 10 years ago after my PhD was like, okay, we find resistant genes everywhere, but first, does it matter? Like to find resistant genes everywhere, is that a problem? And maybe it's not a problem if these genes are just on bacteria in the environment, on the chromosome, bacteria that are very unlikely to be in contact with human or animal, and if they're in contact with this human animal, may not colonize those hosts. But it may be more a problem if those genes are present on bacteria that can colonize the human and or the animals, and even more a problem if now those genes are on mobile genetic elements that are really available to transfer between bacteria. And so that's kind of like the question I was wondering. And to do that, I had to go through the so-called metagenomic approach. So I don't know how familiar you are with those approach, but basically what we know is that if we take an environmental sample and we put it on a plate, there's only certain species that grow, and we're missing a lot of bacteria that do not grow on this plate. So if we want to try to get more an ecological, like an overview of what are the bacteria carrying resistant genes and plasmids, we basically need to go through those metagenomic. However, when we do those, what metagenomic approach does is that we take a community, the total like an environmental sample, and we extract directly the total DNA. And so when we do that, we often we lyse the cell and we break out the DNA get break out in pieces, and often depending if what type of approach we use, but if we use sequencing, we even chop up the DNA in a smaller pieces. And then we can identify markers such as marker for bacteria, or marker for resistant genes, saying, okay, I sample has resistant genes, or marker for mobile genetic elements. And so we have all these kind of studies, all those different approaches that are often used as their own term, where we have also the so-called microbiome studies. Then we have the resistome that consists of the set of all the resistant genes that we can find into an environmental sample. And even some people started to do like what they call the mobilome, which is all the sets of mobile genetic elements you can find into an environmental sample. But what we don't get is, at the end, who are the bacteria, who are the host of the resistant gene and their mobile genetic elements, which has a huge limitation. Oops. What do you do? Okay. Huge limit in understanding the ecology and evolutionary process driving plasmid transmission on this one Earth continent. Okay. And so back in 2014, when I was trying to wrap my head around this issue and trying a different approach, trying to see how I can address this problem, there was 3 papers that came out here and that demonstrated this proof of concept that if you have used in a microbial community, use this technique called Hi-C, you can reconstruct genome in a bacterial community. And I was like, oh wow, that sounds interesting, because so how does that work? And so the idea is that if you take a mixed microbial community and you do a first step of cross-linking, basically you have formaldehyde to your sample that comes to cross-link the DNA. That way, so you have the DNA that is this tiny molecule, that is not this long circle, but more this little molecule all compact into the cell. Then you're gonna have the DNA in close contact is going to get crosslinked. So then when you extract the total DNA, right, then you break out your DNA in pieces, but the fragments that are in close contact stay attached. And so then through a process of a library, you go religate those little ends here of DNA, and so you obtain this kind of like fragment of DNA that are really gated, and you go sequence, and then you can what you can do is you enrich your library for all those junctions to avoid like too much background noise. And then you go sequence each end of these fragments. And so what's gonna happen is that you basically created what is shown here, chimeric fragments of DNA, where each side of these junctions were the same DNA shared in the cell, but not contiguous. So you have non-contiguous DNA in a single fragment. And so if you sequence the end, then what you can do, it can help you to resolve this very complex puzzle of metagenomics, where often what we do is we take the whole DNA of the sample, we sequence everything, and then we try to use like algorithm to reconstruct contigs, right? And then the reconstruction is limited by the number of repetition. A lot of things came that we cannot get like reconstruct full genomes. But what we can do then is like with those puzzles, we can reconstruct saying, oh, wait, here on this fragment, I know this contig and these contigs were in the same cell. And so that way, we can go and reconstruct actual gene. And so I was like, okay, let's try to test this hypothesis. Can we determine the natural carrier of antimicrobial resistant genes and mobile genetic element in a complex microbiome using this technique called IC? And I like to show that has kind of like profiling with the good, the bad, and the ugly, where here in this case would help us to know, okay, those resistant genes are on some bacteria that may be like not be a big immediate problem for transmission to human, or maybe this is kind of environmental bacteria, but it's already mobile, it can move to different bacteria, or even if it's already present, some sort of pathogens that can colonize and infect hosts. So what I did is I went to our local wastewater treatment plant, and I applied wastewater because we know it's a very rich environment of bacteria that carry a lot of resistant gene and plasmids. So that's kind of like, we know we'll find them there. And I tested that. So I worked at that time with a biotech company that was like setting up like we'll say a kit to kind of like help doing those libraries, like trying to optimize the process of doing those libraries. And so for this sample, we on one hand did the shotgun sequencing that we sequenced, and then on this sequence, we went to look for resistant genes, markers for certain type of plasmids, and also another genetic element involved in the adaptation of bacteria to resistance, which are called integrants. And on the other side, we did same sample with this IC library, and then we can use so we use both information to reconstruct metagenomic assembled genome, that's called MAG, and that's what it shows here. So here on the left, I show you just a part of, so we have a tree here where each tips represents a MAG, mostly fragments of genomes. So don't think of the full bacterial genomes. Most of part of the time it's just fragments of genome, but are big enough that we can associate them with a certain type of bacterial phylum. And here I just show you this tree for the proteobacteria, just for simplicity, but we have also much more of a group of bacteria that we found. And on the bottom here, what I show is, this tree and the identification here to the family and to sometime the genesis when we could. And on the bottom, we had all the different resistant genes that we found in this on different contigs, different plasmids that we found, fragments of plasmids, and also integrants. And so what it looked like first is boom, this kind of like a very complex heat map, difficult to read, that where you can see every little square represents an interaction, so a contact, where we could say, yes, for example, this plasmid were associated with this MAG, and the shade of the box represents the strength of the contact that we obtained. And we did an experiment in parallel where we kind of added the bacteria that we know had a resistant gene plasmid, and that helped us to kind of like make sure that we could get the right association where it was supposed to be, and also see that there were some backgrounds sometime where we could find like a noise that will like interact in noise that will like interact with the analysis and help to set a threshold in the analysis. So first, what can we see? Okay, so let's focus first on the mobile genetic elements, which are here, the plasmid and the integrants. What we were happy to find first is that many genetic elements are known to have different host range. And so some plasmids, such as these in-Q plasmids, for example, are known to have the broadest range of hosts, meaning they're like the type of plasmid that can replicate in the most diverse bacteria. And that was interesting to find that we find these plasmid, in our case, also associated to a wide broad range of hosts. And secondly, we were happy to find that many of what we call narrow host range plasmid, like in F, that only can replicate in Enterobacterus, were associated with Enterobacterus. So for us, it was more like a good validation to see, okay, when we use this, it seems that we get the right appropriate associations and be able to say, okay, well, I know these bacteria have those plasmids and these bacteria have, which one have like associated with broad host range plasmid, or also class 1 integrants that were associated with many bacteria on, among the proteobacteria. So that's kind of like the conclusion here. Secondly, then, when we looked at the bacteria and their association, first, if we look at Enterobacter C, as I just said, we found many association with plasmids, which is very interesting, meaning that there's bacteria in those environments carry a lot of plasmid potentially. And when we look at the resistant gene, though, everything you see here is actually just genes that are natural genes in every Enterobacter C that are efflux bound. That doesn't mean they confer a resistance. But the problem here is at that time, when we did this study, the database for antimicrobial resistance were mixing up, like are still like mixed up with sometimes resistant genes that are resistant genes when they upregulated. So it's kind of like very complicated to say, I found this gene, but it doesn't mean my bacteria is resistant. And so here is a little bit in this Enterobacter C, we find all these genes, and that's often in the people looking at resistome we have to be careful between, okay, what's actually just regular function for the bacteria and what's actually acquired. So in the case of Enterobacter C, not much genes, resistant genes, that seems to be acquired. What was more interesting is for other genes. So here I showed one, which is Aeromonas, sorry, at the family level, and that was the family that was the most associated with resistant genes and also broader host range plasmid, suggesting that, and so here's some broader host range plasmid, suggested that this family, I'm going a little bit too fast in my talk because I see I have all the animations that tell you what I say at the same time as I go. So so you can see all these different resistant genes that are found in this family. And so basically, I'll pass and go a little bit faster. That suggested that this family is an important vector of spread of antimicrobial resistance in wastewater treatment. And so we kind of analyzed like all our tree and our associations. And so the conclusion here of the study was that, well, I see helped us identify the host range of plasmid integrant and resistant genes, to reconstruct known associations, that was great, and identify a natural reservoir of antibiotic-resistant genes and mobile genetic elements. I just talked about the Aeromonas C, but we also have the Moraxella C, which is a family hosting Acinetobacter, Bacteroides, and Prevoltel. And I was very happy after we published this study that started to go a bit all in 2019, is that since then, some other people looked with similar approach or different approach in wastewater treatment at potential important bacteria reservoir of resistant plasmids, and Aeromonas always come back as an important one. So that can be an important bacteria in the spread of antimicrobial resistance. Well, that brought me to when we published that, very soon after, got contacted by two ecologists, Alice Risley and Thibault Stalder, who worked on very different things on bacteria, but started to be interested in this kind of relation that between plasmid and host and their eco-evolutionary process driving AMR, and the idea that a plasmid can be a parasite for its host. It's basically, plasmid back and the host live on this what we call parasite altruistic continuum, where in some case where the plasmid confer a benefit, let's say like in the when you have a pressure, selection pressure, such as antibiotics, then you will select, you will have like this plasmid is kind of a benefit for the host. But in some cases, what's been shown is those plasmids can have like a cost for the bacteria, right? It's kind of like an extra DNA you have to replicate. It may like also like interfere with the regulatory process of the cell. So you may have a cost, and in this case, they can be considered as a parasite, and there is all continuum in the middle. And the hypothesis was that beneficial plasmids for bacteria should act as generalists, while antagonists should act as specialists. And that was kind of like their idea here, saying like, okay, their specificities, they work on what they call ecological network, which we may know very well maybe here. And the idea was that, say, if we look at a network of host pathogen or host and plasmids, then an antagonist network will have more modular structure dominated by specialist interactions. While if we have a more mutualistic network, like with interactions, the structure of a network should be more generalist, linking the whole network. And so the idea was to see if we could apply this approach to study plasmid host interactions in natural microbial communities. So this hypothesis, they worked with modeler teams that kind of model this hypothesis that seems to fit. And so what we wanted to do then is like, let's see if we can, with this high-C approach, where we have now a picture of like the association between bacteria, the host, and plasmid, and can we study this like ecological interactions between them? So what we did is like I first like improved first the prediction of plasmid, because I just told you that at that time. So we used the way we detect the plasmid were based on the database that was very poor in terms of detecting plasmids. So we kind of rework on that. We use also like the information from the high ceiling to try to help reconstruct fragments of plasmids. We did a bunch of like basically we improve our bioinformatic pipeline to have a better prediction of what were the resistance, what was the plasmids in this sample, and the association with different hosts, but also which one were carrying resistant genes. And that's what it looked like. It looks just kind of like this network here, where each node, the circle represents a MAG, so a metagenetically assembled genomes, linked to a plasmid. And so the plasmids are represented by the little stars, and when the plasmid is associated with the resistant genes, it's a little pink star here, purple star. And so first, when we looked at this network, and that is colored by class, is at first the network clusters strongly by bacterial taxonomy, suggesting that most of the plasmids are associated with a certain type of taxon. And that's what's also been found in an approach that used database approach, where people have been looking at association of plasmid with hosts, is that plasmids tend to be more associated by the taxonomy in general than what we can imagine spread across all different bacterial species or groups. However, so that's like So for example here, half of the plasmids were associated with less than 2 MAG, and so those could be considered more as specialist interactions. However, few generalists, so few plasmids were overconnecting a little bit the network, and what we observed is that those were also AMR plasmids. And so when we looked at the number of contacts of AMR plasmid with hosts compared to non-AMR plasmid, there were a slight significant more connection of AMR plasmid with the rest of the hosts than non-AMR plasmid. And what Alice did that was very interesting is to compare a sub-network association, where here is a network with only the non-AMR, like the plasmid that doesn't carry any risk associated with antimicrobial resistant genes, the network of the plasmid associated with resistant genes. And so by comparing certain metric associated with those, we can clearly see that there is more cluster in the no AMR condition, less nested, so and more modular network. So the modularity which I put here basically show that there's a high number of separated subnetworks that strongly here is clustered by class. And the fact that it's less nested is basically you have less of those kind of plasmids that come connects the overall network compared to the AMR network. And that was really interesting. That suggested that, and in this case, the AMR plasmid really over-connected, like the had more generalist behavior compared to like non-AMR plasmid that were more like specialist behavior. And when we looked a little bit more into those plasmids that were over-connected, you can see that some were really well over-connected across like beta and gamma protobacteria. So it was basically at the phylum level, with very little interaction between phyla. And one of them, while the others were a group of plasmids that we could not identify, one of them was identified here carrying antibiotic resistant genes as an INCP beta plasmid, which is those so-called rhodopsin plasmid known to be able to transfer and replicate in many different bacterial species. So that's really like conclusion was like these networks are primarily, it seems to be driven by specialism. Does that mean that we have more parasitic role of plasmid in actual environment? Kind of open question a little bit. And a greater prevalence of AMR gene among generalist plasmid that lead to a more connected network that opened the question. That's a little bit the hypothesis we had that were plasmid beneficial in this wastewater environment. We don't know. We haven't measured if this sample had like a lot of antibiotics and there was any selection. But basically, what suggests this result is that maybe there's some sort of selection happening in this environment that make those plasmids maybe beneficial to the host and the community. And more opportunities, it seems that for certain plasmids, for antimicrobial resistance to be connected over the network, so maybe more opportunity for spread of antimicrobial resistance. That led me to this next question, that using those networks, we can also not look at the plasmid host association, but try to understand if we have bacterial transmission of antimicrobial resistance in between ecosystems. So here we just look at one ecosystem. And that's what I'm like studying now. And so we have a PhD student, Marina, that is working on that. So we have several approaches. One of them is, the first is a data mining approach. We were like, well, let's first look in database. There are several databases of plasmids, and so we're gonna look into them. So one is this EMGPR database that's grouped over 700,000 plasmids from metagenomes, metatranscriptomes, single assembled genome or isolates. And one other one is PLSDB plasmid database that is a very highly curated database for plasmids. And so this database has an advantage that here the plasmids are very well curated. We know there are plasmids, they're circular, they're very well characterized. However, they're heavily biased for sampling, their sampling, meaning that most of the strain we find in those databases come from human or animal sampling, and much less from the environment. And that's kind of like here the case. We split this database in two subsets. One, the isolate database that is actually rather similar to the PLSDB, but has different, the genomes came and they have a different strategy how to predict the plasmids. So there are still the difference, but still this bias. And the metagenome database that is interesting here because this database is less biased for sampling because we found a lot of environmental metagenomes. But the problem with this database is the prediction of plasmids, because as I just said, from metagenome, it's very difficult to predict plasmids. So there's tools, bioinformatics tools that allow to do that with more or less success. But here, this database have more shorter plasmids, maybe not full plasmids, and it's very difficult to predict where they come from. But still, it's a very good information. And the idea is to see if we can find common between those different databases. So after filtration for ecosystems that are well identified, host identified, we obtain this about 30,000 plasmids for the EMGPR database, 80,000 here for the isolate, and about 50,000 for the PLSDB plasmid. And then we group those plasmids by identity. So we're basically trying to group all the plasmids that are very, very similar to each other. And so we obtain all those groups. So 8,000 for the metagenome database, 30,000 for the isolate and 12,000. I forgot there's like all those. Okay. And then what do we do? Well, now, as I said, we're kind of trying to identify bacterial hosts that's kind of like in this network, overconnect resistant plasmid across different ecosystems. So what we do is we use this now tripartite network approach, where on the top we have all the group of plasmid we identify. We've in red, the group associated with resistant genes. In the middle, we have bacteria that we group at the genus level, because in the metagenomic base, often database, we do not have like a resolution level to the species. And then we group them by ecosystem, where we have the human on one side, the environment, the animal in the other side, and the environment. And here the environment is pretty broad. We can put everything that was kind of natural environment, but also wastewater, for example, more anthropogenic sample. But we can look also later at other resolution. And so here, what we draw this network, we have here for the EMGPR metagenome dataset, we build a network like that for every dataset. And what is really striking here is you can see that there are, so the middle, remember, is a bacterial genus. And they're organized here by class that are colored. And you can see that there are like focal points that link those certain genes to most of the plasmid described in the database. And some of them are also found in both in the human environment and the animal. And in this case of this database, interestingly, we don't know why yet, but with all after our filtration, unfortunately, the animal side was not very well represented. But for the other database, we have the animal well represented, the environment less represented. And so the question then is like, okay, among those data sets, so 1, 2, and 3 data sets, then what we did is to try to identify which bacterial genuses were consistently associated with resistance plasmid across the 3 types of ecosystems. And those are called the core bacterial genus. But also, like, we left a little bit of like a room also for what we call common bacterial genus that will be found across the 3 ecosystems in one data set, but only 2 in the other data set. I'm not going to extend on those common and really focus on the core first. And so that's what shows here on the right. And so what we found is that there are 19 common bacteria, 9, I've gone wrong. The core is here on the right here. I was like, what is? So we find 9 bacteria across 9 bacterial genuses across the 3 databases that are consistently associated with resistant plasmid across human environments and animals. And so that's what we showed here. So it's a little bit heavy to read, not an easy slide, I would say, but what shows this slide is all the basically core and common bacteria type that we found. And in the ones that are darker are all the core, so bacteria found across the 3 ecosystems. And the one highlighted in red are the ones associated with antimicrobial resistance plasmids. And so what we found Acinetobacter, Enterobacter, Escherichia, Klebsiella, Pseudomonas, Salmonella, Serratia, Staphylococcus, and Streptococcus as genus that are often very associated with resistant plasmids across the animal environments and use. And so what is interesting is those genuses are all like genera hosting SKP pathogens, which kind of like are often generalized group of bacteria that can live in multiple different environments. And so that suggests that maybe there's cousins or those escaping pathogens play an important role in the spread of AMR across the human environmental animal continuum. And so also, are those groups have like more like spreading specific strains with more AR plasmid than others? That's kind of like what we want to identify using different approach of isolating bacteria or by using this high-C approach I presented to you earlier. And another thing that is important is because still we use database, and the database are heavily biased for SKP pathogen because they tend to be more known, more sequenced. Even though in the metagenome database, there's less sampling bias, still like the predictions, all the algorithms that are used to predictions are often trained on what we know, which are more like SKP pathogens. So there's always a little bit of limits in our brain of like, okay, it's very interesting, it's maybe real, but we may have like some bias. And so that's why we want to do the same with this experimental approach. Where now, so we want to go across, so we're going across different environments, and human-associated environments, animal-associated environments, and natural environments, and using this high-C approach I just talked about earlier. But now we're adding another step. So what we do is this target capture step. So the idea is that we created this chimeric DNA that is shown here, and the problem is that when we sequence this library, most of the DNA are chromosome bacteria. And what we are interested in is like plasmid or resistant gene associated with chromosomes, and those associations are often not dominant in those libraries. In addition, that often bacteria found in the environment to carry antimicrobial resistant plasmid are not the dominant bacteria. They often present at rather low relative abundance. And so that's why we are including this now like new target capture, where we target all the resistant genes that we know, basically, and we go find in our library and reach for those resistant genes, and then trying to find the the host of resistant genes. So that's ongoing work. We're still setting up the approach, but this approach, I don't know the time I have. Yeah, I was not sure. That's why I was going to stop here or not. But basically, this approach comes from like another study where we were interested in understanding whether or not plasmid transfer in the rhizosphere of plants. So when we apply manure on soil and we have bacteria from the manure with resistant plasmid, those plasmid can transfer to soil bacteria and create a new reservoir for a long-term reservoir for those resistant plasmids. And so we wanted to see if that can happen in the rhizosphere of plants that is suspected to be a hotspot for horizontal gene transfer. And so what we did, that was kind of a work from Chava, like the grad students that worked with me. And so the idea was here, exactly what I just said. We have like a bacteria from animals and bacteria from the environment, and then we have those interfaces here where we have manure that is applied on soil, and we have bacteria from one ecosystem meeting the other, and do we have a transfer of plasmid from one type of bacteria to the other? And we know those transfer events happen at very low frequency. Like in the 90s, many people were trying to add bacteria in soil and trying to measure frequency of transmission. And usually they're like 10 to the -5, 10 to the -7. So it's very low frequency. And because there's not strong selection to go for enrichment then after this transfer event, we often don't see those events they're very difficult to detect. And we know that with the metagenic approach, it's kind of finding this needle in the haystack. And that's where comes this idea of using kind of a magnet to try to find the needle. And so what we did in the first study is set up this approach where we use the idea, the IC approach, right? To say, can I detect in my microbial community, if I give a bacteria with a resistant plasmid that I know, can I find, using this approach, IC, a new recipient here in this purple bacteria? And so to do that, we design specific probe to this plasmid. So in our library, we can go find these specific fragments. And then the idea is that we only get plasmid contact, which we don't care. But what we care is the plasmid host contact, and eventually those blue ones where we find a plasmid fragment associated with the new chromosomal host fragments. And so then we can sequence that and search for new hosts. And so that's, we kind of set up, look at the detection limit of this approach. And so what we did is we took in a microcosm experiment, we added realistic amounts of manure where we spike in an E. coli with a multi-resistant plasmid PB10 to a realistic amount also. And we added that to soil, and we waited 30 days, which is the amount of time we require before seeding a plant. And then we seeded our plant, we grew our barley in pot for 3 months, we harvested our barley in the field, and then we continue monitor the pot for an extra 3 months. And we carried out these experiments both with the E. coli with PB10 and control experiments with the E. coli without PB10. And so what we did at first is monitored our donor bacteria, the E. coli with the PB10, and by plating on the plate, and you can see that still, like, we're impressed how long this E. coli can stay in the soil for at least 2 months. So it's kind of there's a decline. Here we see that the 0 is when we seeded the plant. And there was a little, maybe we don't know if there was like a little bit increase or not that could be linked to the growth of the plant and the rhizosphere bringing nutrients. And we did in parallel the same, but with ddPCR with probe. And primary probes targeting specifically the E. coli, and primary probes targeting the PB10 plasmid. And so the yellow is the host, this E. coli, and you can see again a decline, a little increase, and then like suddenly the host kind of like went below our detection limit. But for the plasmid, what we observe is that we detect the plasmid all along the way, suggesting that maybe the plasmid transferred to a new host. But also, we don't know, it could be like just plasmid maybe in the soil. There's like, we don't know how long plasmid could stay like in the soil like that. So that's where like this high C approach could be like, was used to trying to find if we can find a new host. We did plating also to try to catch new transconjugates, so new bacteria, and nothing grew. So we couldn't grow anything with that plasmid. And so what I show here is for those time points, we did this technique, high C plus, now we call, because we added the target capture. And so what we found here, for example, at day 10, where we still detected our donor by plating and PCR, is we looked at the association of reads that are the E. coli with the plasmid, so the donor, and we find them here nicely. And we find also the PB10 on itself nicely, and we find here reads, many reads that are actually our PB10 with underlined. So when we say underlined, it's not underlined to the E. coli, suggesting it's a new host. However, beyond this time point, despite the fact that we detect those reads underlined with new hosts, you can see that our control experiment, we also detect those reads. And you're like, well, why did you detect like the E. coli, basically, like unknown, sorry, the E. coli, the red ones, what do you detect, sorry, like PB10, not the E. coli, PB10 reads in pots where you haven't added the PB10, right? And the answer is that in soil, especially in the rhizosphere, there is a lot of natural plasmids that are naturally occurring at very low abundance, and actually are cousins of this plasmid PB10. And they have sharing some sequence. And when we add those probes, they're always going to try to bind to something. And so if they cannot bind to your target, they're going to bind to what's the closest. And so what happened here is that we enrich also for background of plasmids that are naturally present in the environment, which was a problem here, because then we cannot say whether or not what we detect in this PB10 are really our plasmids that transfer to a new host or not. But in this one, yes. This one we're pretty confident that's what we detect. And the problem is, so we find this evidence that we have a transfer after 10 days in the planting the barley and not later on. But the problem is that when we looked at the reads that are associated with this plasmid, those new reads, we could not identify those. When we say could not identify, it's like this DNA doesn't map anything in databases. So when we map it on databases, it just comes out as like mapping nothing. We were worried maybe it's like a sequencing artifact. We're trying to like look more closely. No, it looks like really like no sequencing artifact, and it seems to be DNA from the host. We don't have a lot of them to be able to reconstruct a genome. We have like a few thousands, but still like nothing. So that suggests that maybe those new plasmid hosts are likely maybe unculturable members that have been like culture grow, or also detected by classical metagenetic approach in soil microbiome. Because remember, we do the target enrichment, so those bacteria may be just present at very, very, very low abundance in general. What is interesting is that another paper used a different approach with kind of different goal, but similar result is that in the experiment, they only observed plasmid transfer in the next few days after you see the plant and the plant grow on the rhizosphere, they can detect this transfer event, and then after that, they couldn't detect anymore. So if it happened at too low frequency to be detected. On that, I'd like to thank you. So the work that I presented that was done when I was at the University of Idaho, and now like the team where I'm working recently in English. So thank you very much.
Thank you, Thibault. And floor is open for questions, and also for the people online, you can leave your question on the Q&A, please. Yes.
Thanks a lot. Maybe I can start. So I had a question about, is it in English or French?
It's in English.
In English, okay. About how you chose your environmental samples and animal samples, particularly animals. Were they farm animals or were they wild animals?
So you talk about when we do the network, like
Yes.
So this is from database. Like the first work we did is from database, so we don't pick where they come from. But the study we're currently doing, where we taking samples, we take farm animals. So like near Limoges, we have a lot of cows, so we like focus right now on cow farms where we can have access to the animal and the environment. And then for the environmental sample, we go from basically the sink all the way to the river across like the wastewater treatment. And we may include a little bit of human samples. Those are will be biased in the way that we look at a patient sample hospital.
So the environment is the river. It's not have soil.
No, we do. We have the soil. Like with the environment is when I call environment is there is both what I call wastewater is environment. We have river water and soil also. Sorry, soil sample from the farm where they apply the manure on the top. So we have soil samples that are like with different rates of like treatments, and also the environment of the farm, and it's like the drinking water, the hay, also the peat or the straw on the ground.
So it's a little bit like this is driven that, for example, manure is contaminated.
Yes.
The soil is.
We don't have too much like a very very natural environment, because here we're trying to find a little more connections. So where we can find maybe those bacteria that are those hubs between the transfer.
And so that comes to my second point. So you mainly focus on non-antimicrobial resistant genes.
Yes.
Would you capture some animal ones, like by lowering maybe the conservation thresholds, or what are your thresholds for you?
It's a good question. So when we do the isolate approach, we're like trying to isolate based on ECOF value for different bacterial genera, and trying to capture which one require new resistances. Right now, I don't know if we will capture new resistance or not. Potentially, yes. It's not like the goal. I don't know if we'll find. Maybe we'll find like a plasmid that confer resistance, but we don't know why there's a new gene on it. But people have been doing that also. I've never heard about finding new resistant gene that way, but maybe. And when I do the methodological approach, I try to be very conservative. And so I try to be at least more than 90% identity to a gene, because I don't want to lose. I want to make sure we're talking about a resistant genes and not something else. And this is not the goal here. Like I don't use too much of a noisy threshold. One thing that's very interesting in the sample, it's a totally different study from the team from the INSERM that we're finishing, is using a totally different approach based on these integrants or platform acquiring new resistant genes and allowing bacteria to express the gene. Is that when we look through a continuum like that, we find a ton of new resistant genes. We find new resistant genes, especially new resistant genes to beta-lactams, that are really seems to be enriched in hospital electrodes.
I think this is an important thing. If we want to identify some new methods of capture, a good thing.
Yeah, it's true. Here we're looking more retrospectively and not aiming to predict maybe what will be the future resistant genes that will show up in at hospital.
Yeah, I had a question about so you are active bacteria that are playing the nerves for the disease, but I was wondering how you control for also the effect of the abundance, the relative abundance of some bacteria. So are they really more generalist, or I mean, it also applies for the persons, are they more abundant? So how do you disentangle it?
Yeah, so for the abundance, so again, on the part that is database driven, we don't know like the abundance much of those bacteria there. Like we can look at the abundance with the abundance of what's found in the database. On the isolate parts, we definitely know that they tend to be not very abundant because when we don't use selection, we found most of the bacteria to not be carrying resistance, but as soon as you add a little bit of resistance, you find them, so they're not like the most abundant. And on the metagenomic approach, again, because we're using this targeted approach, it's difficult to go really have a good sense of the abundance. What it seems to be is that often what's the more abundant get enriched the most, and that seems to be rather linear. But I would not put my hand like to say like, yeah, this is what's the most abundant. So on that note, we cannot say, we can just say there are bacteria that we find across multiple environments. And what I want to see is like they test their capacity to metabolize also a lot of different carbon sources to show that those bacteria are able to grow everywhere, but not really on the abundance. Yeah.
So I see. Does it allow you to estimate the copy number of the plasmids?
Can we get the copy number? No, again, you could, because you always have reads and you can map them back to your strain. The problem in the microbiome is that you don't know if this plasmid is 1, 2, 3, 4. So on a single strain, when we do genomics, yes, we can estimate the copy number. When we do like the PCR, we see that our PP10 is present at 10 copy per cell. And what we did with the high-C, actually, yes, we were able to predict actually the abundance, the frequency of plasmid in the community, but that's independent of the copy number. So no, I'm not going again with the copy number. You could still try, but I don't know if it will be very accurate.
I have one quick, yes.
I have a very general question that goes back to one of the first slides that you have, where you have the different ecosystems and the arrow going from individuals or farm environments to all the other environments. Is this the way it goes? So the arrow goes one way. Does the resistance always start in, like me, when I take antibiotics, or me feeding the antibiotic in environment, or a cow feeding the antibiotic and following?
That's $1 million question.
When did you start in general?
Yeah, we was egg. So it's a little bit of everything. It's I would say there are cases. I go all the way back, but there are cases where we carry our bacteria that are going to be problem later. So some bacteria are just naturally carrying those resistant bacteria. They don't give you any issue right now. So most of the bacteria that give problem resistant at hospital are what we call opportunist pathogens. They're not pathogens. You may have them on you. They're not giving you any problem, but when your immune system go down, that's where it's a problem, because they can colonize a place where they're not supposed to be, whether it's blood or another part of your body. And that's where like, you get an infection, your body, if it's the immune system too low, cannot deal with it, and that's where you need to take antibiotics. But if they're resistant because they acquire a resistance, that's like the problem. And some others we know are carried by, we carry them. Some others we know are acquired from the environment. And I think the most example that have been the most described, I would say, on terms of from the environment to the human is a hospital sinks trap. And there are cases where it's really well documented that sink trap at the hospital can be a source of infection with resistant plasmid, even not the strain. So somehow resistant plasmid found in the sink trap went to a bacteria on the patient, or not another bacteria somewhere we don't know where, the patient get infected with that. And there are reported events where sometimes patient comes and carry the resistant bacteria, inoculates maybe the sink trap, that stays in the sink trap. Several months later, another patient comes to the same room, get infected by the same plasmid. So there are both directions. So, and that's what we don't know is the weight on those arrows I was showing. And this is kind of very complex. You have, like I said, and you have the animals, so environment, and we're trying to modelize that a little bit where we put arrows a little bit everywhere, but we have no idea of the weight of those arrows. That's something we love to try to understand a little bit, yeah. But so a little bit everything. That's my answer.
I think there is no question online as I see. Maybe one last question and then.
Yeah, sorry.
No problem.
In the menu, you have, it's known that the Enterobacteriaceae are decreasing. So somehow you are selecting if it's decreasing, it's degrading, right?
Yeah.
You're selecting somehow the plasmid that will not be transmitted, or because what do we know about these plasmid carrying resistant genes that are present in Enterobacteriaceae and that are transferred in other bacteria that are staying in the menu?
Well, that's what we don't know. That's what we're trying to see a little bit like. So we know the resistant genes in the manure decrease, and on soil it tends to kind of like disappear out of detections. What we know is that it seems that repeated application of manure over and over time, you have a faster increase of the resistance genes in your soil in general, and you may have eventually have this reservoir over like 5 to 10 years of plasmid, of resistant genes, suggesting that there's been an implementation of those genes somewhere, but we don't know. Like,
and PB10, how do you know that it's still functional? Because you detect
No, we
it
don't.
by PCR. But
yeah, exactly. So I say like the PCR was just kind of here to tell us.
but you don't know if it's
That's why like the high-C is here to kind of like, okay, like at day 10, for example, like we capture new fragment of DNA with the plasmid that are suggesting that these were in a cell at some point. But no, the PCR approach, no, it was just to give us an idea, like it's possible that the E. coli is still there, just under the detection limit and detected by PCR. It's possible that, let's say, the plasmid is free. Maybe there's been people that have shown that plasmid that can stick to soil particles and stay for a few days or weeks, and that fragments, yeah. And so that's possible, and that's why like the PCR doesn't tell us anything. It just like give us an idea where we're gonna get our sample to use with the high-C to try to find them. But no, it doesn't tell us anything. So the only thing we can be sure is say after 10 days, we're pretty confident we had a transfer event, and that's all.
And before we thank Thibault once more, just a small announcement. We're going into a long break now with all the French holidays coming up in May. So we will resume, if I'm not wrong, on the 22nd of May. Thank you very much.